So we're going to talk about enzymes today, and hopefully this will be a little bit better than some of the other videos. We shouldn't have the camera focusing problems. So we're going to start first with some characteristics of enzymes. Okay, one of the first ones being that they're a protein. Remember, we've done proteins as we have done, as we did organic compounds and macromolecules. Remember, a protein is a polymer. It's a long chain of amino acids. It's got a wide variety of characteristics, and one of the examples we gave of a protein, we did organic compound, was an enzyme. So an enzyme is a kind of protein. It is also what we would call a catalyst. And a catalyst is going to be anything that speeds up a chemical reaction. Oh, I am not very good at writing on this yet. So a catalyst is going to be anything that speeds up a chemical reaction. So enzymes are catalysts. That's what they do. They speed up chemical reactions. Some of the other properties of enzymes are that they are reusable. So once an enzyme binds with a substrate and activates the substrate, speeds up the chemical reaction, that enzyme can then be reused. It is not consumed during the chemical reaction. Enzymes are also what we call substrate specific. We'll look at a picture of that in just a minute. But then when we're talking about substrate specific, that means that the enzyme can only bind with certain molecules. Enzymes are very picky about what they will bind with. And also, just a kind of a random note, kind of like we had with sugars and carbohydrates, how they ended in "ose." Your enzymes are going to end in the suffix "ase". So again, when you're taking a test and you see a word you don't know or you don't recognize, and it ends with this "ase" suffix, you need to think that that may be a particular kind of enzyme that is doing whatever chemical reaction it is that you're talking about. Remember, enzymes will end in "ase", sugars will end in "ose". So let's talk a little bit about the enzyme's actual structure. So the enzyme will have what's called the active site. And the active site will be this area right here. That's where the substrate is going to actually bind to the enzyme. So the orange here is the enzyme. So the orange molecules here, these are the enzymes. And the active sites, again, are going to be these spots here where the substrates will bind to them. So these molecules here, these are going to be your substrates up here. And so these molecules are what will fit into the active site. And you'll notice here, you can see how these enzymes are substrate specific. The circles only fit into this enzyme here on the left, and the square, the rectangular shaped ones, they only fit in the enzyme there on the right. That's what we mean by the enzyme being substrate specific. Only certain molecules can fit into the enzyme. We're going to be using both of these terms quite a bit throughout your labs and throughout your tests. So active site, we do need to be very comfortable with that is. So the active site is on the enzyme. And the substrate is the molecule that actually binds to the enzyme. So the substrate is not part of the enzyme. It is the molecule that binds to the enzyme. So what we have here is a picture of induced fit. So we were talking on the previous slide that that enzymes were substrate specific. They would only bind with certain substrates, and that is roughly based upon their shape. And so as we look at this picture here, you'll see this enzyme has a loose shape that goes with this T-shaped and triangular shaped substrates. It is roughly shaped the same as they are. And as the first substrate comes and binds to the enzyme, you'll notice there's a conformational change in the enzyme. This enzyme, the active site on this enzyme now, looks a little bit different than it did initially. And so there's been a conformational change to the active site. And so now these substrates do fit perfectly into that enzyme. And so that is what is called the induced fit model. That the enzyme will have to change its shape slightly to conform and to form a nice tight fit around the substrate molecule. So that active site will shift a little bit and form a nice glove, really tight fit around those substrates that bind there. So let's talk a little bit about what the enzyme's job is, what its function is. Okay, so the job of the enzyme is going to speed up chemical reactions, and it's going to do this by lowering the activation energy. Okay, activation energy is going to be the energy that is required to get the chemical reaction going. Okay, and so our activation energy is going to be higher in chemical reactions that do not have enzymes. Okay, so this activation energy, again, if we look at this graph here, we can see our activation energy without the enzyme is this red line. Okay, and so this red line here, okay, is indicating our activation energy without the enzyme. And you'll notice it's significantly higher than the black line, which is our activation energy with the enzyme. Think of it as you're running a race, okay? And the actual race is the chemical reaction. And to get to the race, okay, so to get to the actual start of the race, you would either have to run over a mountain, which would be this big red one here. You'd have to run over a mountain, or you would have to run over a hill. Well, which one would you rather do before you're going to start running a marathon? Would you rather run over the mountain or the hill? And so this will allow the chemical reaction to happen faster. Just like if you were running a race, you would be able to run your race faster if you just had to run over a hill to get started, as opposed to run over a mountain to get started. So when we lower the activation energy, that's going to speed up the chemical reaction. And so that's what an enzyme's job is. And that's how it's going to speed up the chemical reaction. It's going to take that activation energy and it's going to turn it from a mountain down into a hill so that that chemical reaction can happen faster. And so there are two types of chemical reactions that the activation energy can have an impact on. There are what are called anabolic reactions. An example would be dehydration synthesis. Okay, if we kind of break this down a little bit, when we synthesize something, okay, we are making something new. Okay, and so if I synthesize something, I am making something new. So that's what my purple molecule is there. Okay, so in this reaction, this is an anabolic reaction. So I have built something new. I have taken these two substrates. They have bound to the active site on the enzyme, and they have joined together to form a new product. And again, an example would be a dehydration synthesis. So a dehydration synthesis would be when I formed a new molecule. Okay, in addition to forming a new molecule, to form that molecule, I released water. That's why it's a dehydration synthesis. Okay, so in forming that new molecule, water was released. The other type of chemical reaction that can happen is the opposite, okay, which is called a catabolic reaction, okay, or hydrolysis reaction. Sorry, my box shrunk on me somehow or something. I'm not really sure what happened. Okay, so the catabolic reactions, they're going to break things down. An example here would be a hydrolysis reaction. So in this picture that we have down here, we have this substrate here, okay, that's headed into the active site of the enzyme. And when it leaves, when the enzyme releases it, it actually now has broken it into two substrates. So if y'all think back to some of your vocabulary, you know that lysis means to burst or to break apart, okay, and hydra would be water. So an example of a catabolic reaction would be a hydrolysis reaction, which is actually the opposite of your dehydration synthesis. So with your hydrolysis reaction, you would add water, and water coupled with the enzyme would help break that molecule down. Okay, so we've got anabolic reactions that build things up, okay, or synthesize things, and we've got catabolic reactions that are going to break things down. Okay, and we've got two examples of those happening here. The last thing we need to talk about here are things that affect the enzyme reaction rate. How fast can this enzyme do its job? So one of the first things we're going to talk about here is temperature. Okay, and so you'll notice on your graph here, you've got your enzyme activity over here on your y-axis and your temperature over here on the x. And you notice there's an optimum temperature. Okay, right here around 40 degrees Celsius, my enzyme activity is as high as it's going to get. Okay, right there around 40 degrees Celsius. And then you'll notice after 40 degrees Celsius, there's a very sharp decline. Okay, very rapid decline in enzyme activity. Okay, and one of the things we need to be aware of with temperature is something called denaturation. Okay, and with denaturation, okay, the enzyme actually loses its shape. If we think back to the very beginning, okay, one of the characteristics of an enzyme is that it is a protein. And the protein's ability to do its job hinges on its structure. Okay? Protein structure and function go hand in hand. And so if a protein is a different shape, then it can no longer do its job. And so an enzyme is a protein. And when the temperature gets too hot for an enzyme, like it's doing over here in this section of the graph, when the temperature gets too hot for the enzyme starts to lose its shape. And if it loses its shape, it can no longer work. Okay? And that's what denaturing is. If an enzyme denatures, it loses its shape, and so it no longer works. And too hot of a temperature is a time where that could happen. Another thing that can affect our enzyme reaction rate is the pH level. This is our acid-base level. Just like temperature, there is an optimum pH level, okay, and that will coincide with our optimal rate of enzyme activity. Okay, and then when the pH is too acidic, the enzyme doesn't work very well. And when the pH is too basic, the enzyme doesn't work very well. And enzymes will vary from place to place. Enzymes you have in your stomach would have an optimal pH way over here, okay, very acidic. Okay, enzymes that are in your body in other places than in your stomach would have the optimal pH more there in the middle. So optimum pH will vary from enzyme to enzyme. Another thing that will affect our enzyme reaction rate is the concentration of our substances. Okay, remember we're dealing with two different substances here. We've got our substrate, which will bind to the enzyme, and the enzyme itself. So let's look at substrate concentration first. Okay, what I have here is on our graph of substrate concentration. So down here at point A, my enzyme rate of reaction is increasing very quickly because there's very little substrate concentration here. And I've got plenty of enzyme. And remember, enzymes are reused. So as soon as they finish with one substrate, they pick up another one and just keep going. But you'll notice right here, okay, I hit a point, okay, where my enzyme rate of reaction levels out. Okay, you notice it doesn't go any faster. And when it reaches that point, okay, it's what is called saturated. And so what we have here is that there is enough substrate for every single enzyme. Every single enzyme that is present is busy. Okay, it doesn't matter how much more substrate you add. So as I head over here to part C, I've added more substrate here in part C because I've increased my substrate concentration. So I've added more substrate there in part C. But at this point, it doesn't matter how much substrate I have. Way back here at point B, every enzyme became busy. And even though they are constantly working and they are reused, okay, there just isn't any more enzymes. They cannot go any faster. They're at their maximum rate. Okay? So in addition to substrate concentration, enzyme concentration can have an impact as well on the reaction rate. Obviously, if there are fewer enzymes, your reaction rate will be slower. The more enzyme that you add, the reaction rate would speed up. And the last thing we're going to talk about here is our last thing that can affect our rate of reaction for the enzyme, and these would be inhibitors. Okay, and inhibitors obviously inhibit. So when we inhibit something, we stop it from happening. Okay, so these inhibitors stop the enzyme from being able to do its job. So all inhibitors are going to have a negative impact on the reaction rate. When inhibitors are present, your reaction rate will slow down. Okay, it doesn't matter what kind of inhibitor it is. So I've got two basic kinds of inhibitors. I've got a competitive and a non-competitive inhibitor. So a competitive inhibitor, let's erase that. I thought I was going to drag my picture out. Okay, a competitive inhibitor, that's one where the enzyme is going to, the inhibitor is going to bind directly to the active site. You can see up top here is a normal enzyme function. Here's my substrate bound to my active site. Okay, and it releases these two products. So that would be a catabolic reaction, okay, possibly a hydrolysis reaction if it released water with that. When I have a competitive inhibitor present, okay, you'll notice my inhibitor is bound to my active site. So my inhibitor looks an awful lot like my substrate here. Okay, it's the same, roughly the same shape, because remember enzymes are substrate specific, so it's roughly the same shape, and so it can fit into that active site. And so now the substrate can no longer get into that enzyme, so that's going to slow down our rate of reaction. Okay? Our other kind of inhibitor would be a non-competitive inhibitor. So a non-competitive inhibitor, you'll notice that one is binding to somewhere other than the active site. So the substrate, the active site is still available for the substrate, but if you look at the active site when the inhibitor is present, the active site does not fit the substrate anymore. So a non-competitive inhibitor binds somewhere other than the active site, and it changes the shape of the enzyme. So now the substrate doesn't fit into the active site. So competitive inhibitors are in direct competition for the active site. Non-competitive inhibitors still slow our reaction rate and make our enzyme no longer functioning, but they did it by binding somewhere other than the active site. And so now the substrate cannot fit. Okay, so we will do some enzyme modeling in class next period, and then we will be prepared to do our enzyme lab after that.